Hello and welcome to Chapter 3, Cell Biology. This chapter is a lot like Chapter 2 in that it's just giving you an overview of some of the important concepts from biology. If you recently took a biology course, most of these things, won't be new to you. If you took biology in high school, you most likely learned about all these things. But again, what I'm going to try to do in these lectures is connect them to A and P and kind of emphasize why some of these things will be useful for you to understand when we get into some A and P topics. But hopefully, again, this is just a review. It's been a while since you took biology, you might have to spend a little more time, but this chapter shouldn't be overwhelming. But again, hopefully it'll help you understand some of these biological concepts. So let's start with our list of topics. First one is cell and its cell structures. So we're going to talk about the plasma membrane, basically what forms the membrane, which in the previous chapter we learned that it's made up of a phospholipid bilayer. It also is going to have some different proteins in it, as well as if you're an animal cell, you have cholesterol in the plasma membrane. We'll also kind of give a quick overview of the organelles. Again, a lot of these organelles you've probably heard of, such as the nucleus, mitochondria, endoplasmic reticulum, and a few other ones. We're going to spend quite a bit of time on the membrane, the plasma membrane. That's an important structure because it determines what can go in and out of the cell. That's where you might have receptors for hormones or neurotransmitters. And, we're going to be talking about hormones and neurotransmitters in A&P, so it's important to understand, where those would be located. So we're going to talk about different membrane proteins and their functions. We're going to get into membrane transport, which is, how do things enter the cell? How do things exit the cell? A lot of times it's using membrane proteins. Sometimes we call them channels or transporters or pores. But if you don't diffuse through the membrane, you're going to need some sort of membrane protein to help you enter or exit the cell. We're going to just quickly go over transcription and translation again. We did touch on that in the last chapter, so we're not going to do that much more than we did in chapter 2. But again, I want to remind you of those two processes. And then we're going to wrap it up with a little bit on cell reproduction, mitosis and meiosis. And just so we're clear as to what those two processes result in and some of the differences between those two. Okay, so let's begin. Every book's got this picture one way or another. So it's probably something you've seen in the past. This is your kind of prototypical animal cell, although not all cells look like this. We're going to learn about a lot of cells that don't look like this at all. For example, a skeletal muscle cell doesn't at all look like this. They're long and cylindrical, and they have a lot of nuclei. They don't just have one nucleus. They actually have multiple nuclei. So not every cell looks like this, but this is kind of your prototypical, cell to kind of start from. But you'll notice that it's got a plasma membrane around the outside. You can even see in this little circle here the phospholipid bilayer and then a little protein embedded in the membrane. Nucleus, we usually start at the nucleus. You'll see that the nucleus is going to contain, a center area called the nucleolus. But for us, the most important thing is to know that that's where you're going to find your chromosomes. And we should know how many chromosomes are in a human cell. That number is 46. If you're a somatic cell, somatic means body cell. So if you're any of the body cells, skin cells, muscle cells, neurons, you have 46 chromosomes. If you're a gamete, which is a sperm cell or an egg, you're going to have half that number. You're going to have 23 chromosomes. We refer to the 23 as haploid and the 46 as diploid. And again, we're going to revisit those terms at different points in our A&P. You'll notice kind of around that nucleus is the ER. The ER has two parts, smooth and rough, and we'll kind of talk about the difference as we move forward. The Golgi apparatus is shown here in green. It's going to actually receive a lot of molecules from the ER and then ship them out. So we'll talk a little bit about that. A lysosome, that's a little membrane enclosed organelle there. We see multiple mitochondria. Mitochondria is plural, by the way. Mitochondrion is singular. We see some cell extensions out here. We'll talk more about those when we get into the tissue. Oops, sorry, my dog just touched my computer here. We'll talk more about these microvilli and cilia in chapter 4, but some cells have these cellular extensions from the membrane. Okay, this is kind of a redundant picture, but you'll notice these are some of the main organelles we want to be able to know and understand their functions, which is given on the next slide. So here is a little table just describing the key functions for these cell structures. So plasma membrane, again, phospholipid bilayer, some proteins in there, cholesterol's in there. It acts as a barrier, but a semi-permeable barrier, which means it's going to allow some things in and out, but prevent other things from going in and out. And it all depends on the cell type, and it depends on, depends largely on what kind of membrane proteins that cell has. The nucleus, again, contains chromosomes, 46 chromosomes for most of our cells, 23 for sperm and egg. Ribosomes, if you remember from translation, they're the ones doing the translation, so they're taking mRNA, kind of deciphering the information in the mRNA in order to make protein. So protein synthesis. The ER, two types of ER. You have the rough ER, and it's rough because of the ribosomes. So if the rough ER has ribosomes, then the rough ER also synthesizes proteins. And then the smooth ER does not have ribosomes, but instead it's going to synthesize lipids for the cell. It might store calcium. That's going to be a very important function when we get to muscle. Muscle needs calcium in order to contract, and it needs to store it and then release it. And that's done using the ER. If you're an organ like the liver, which has a very important function in detoxification, that's done using the ER. Golgi is a lot like a UPS store. So it's going to receive things from the ER. It might modify them a bit, change them a little bit, repackage them, and then ship them out. So that's the function of the Golgi. The lysosome has enzymes that can break molecules down, kind of digest them, if you will. So the lysosome kind of will receive maybe molecules that aren't working anymore or organelles that don't work anymore, and it'll break those things down so we can reuse the parts. So it's almost like a recycling organelle. Lysosomes are going to be important when we get to the immune system. My dog is making noises. The lysosome is going to be used during a process called phagocytosis. So that's where you have an immune cell that can basically engulf a microbe, like a bacterium or a virus, and then in order to kill it, it uses the lysosome. It uses these enzymes that destroy the microbe. Finally, we have mitochondria. Again, you probably have heard, it called the powerhouse of the cell, but we're in college now, so we need to take it a little further than that. It's the site of aerobic respiration, which is where the cell makes most of its ATP. So those are your basic functions of these important organelles. Plasma membrane. So here's our drawing of a membrane. Again, you might have seen this in the past, but you have the phospholipid bilayer. Those phospholipids have the nonpolar fatty acids facing one another, and then the polar phosphates on the two surfaces. So you'll notice this side they call the external surface. This would be the inside of the cell, so this would be the internal surface. So the phosphates are on each surface. You'll notice embedded in these fatty acids is cholesterol. So cholesterol is a requirement for animal plasma membranes. It's not found in plant cells, but it's found in animal cells. Then you see these different types of proteins. These little colorful things are different proteins. You see this one acts as a channel, so it might allow something in or something out. Some of these proteins have little sugars attached, so they're known as glycoprotein. Glyco means sugar, so it's a protein that has little carbohydrates attached to them. This happens to be a lipid that has sugars attached to them. This one they call integral because it's kind of embedded inside of the membrane. Others are known as peripheral because they're kind of attached to the surfaces. So that's your basic components of a plasma membrane. You'll also see on the inside there's the cytoskeleton, so those are proteins that kind of form a skeleton for the cell. But that's your basic membrane protein. And what we're going to talk about are these proteins here and what they might be doing for that cell. So membrane proteins. These are going to be extremely important when we get to how neurons function. So when we get to the nervous system, I'll remind you about ion channels. So ion channels are how ions go in and out of cells. Because ions have charges, right? Positive charge or negative charge, they can't go directly through the membrane. All right? Some molecules can. So typically, if you diffuse through the membrane, you're typically going to be nonpolar, all right, because you got to get through those fatty acids, and you're typically going to be small. Ions are not nonpolar because of that charge, so they can't go through the membrane. So if you're going to move sodium in, or potassium out, or calcium in, you're going to need ion channels. All right, so this is just the basic drawing. These little spheres could be some ion. Let's say it's sodium, and this sodium is going through the channel into the cell. Other membranes might be more what we call a carrier. Really the only difference here is carriers change shape. They kind of have to grab a hold of the molecule, change shape, and then almost spit it out on the other side. That's how a lot of our amino acids enter cells. Other proteins might be receptors. So we'll talk about receptors in the nervous system, as well as receptors of the endocrine system, so hormone receptors. So basically a receptor is like, how a cell responds to a chemical, right? How does the cell know that that chemical is there? It's kind of like having a doorbell, right? How do you know someone's at your front door? Well, they have to ring the doorbell. In this case, how do you know that chemical is there? It has to attach to a receptor. And then that usually will stimulate that cell. It'll get that cell to do something. All right? So membrane proteins could be receptors. They could also be enzymes. So an enzyme is just going to cause some sort of chemical reaction. This would be a membrane-bound enzyme. And it's taking this molecule they call a dipeptide and breaking it into amino acids. So that's just one example of what an enzyme could be. We're not going to really talk too much about membrane enzymes. Linkers. Linkers just will attach the cell to maybe a protein outside of the cell. This is often done so the cell could potentially, be stabilized, right, to kind of hold the cell in place. A lot of cells need to be, attached to other tissue, so they'll use these linker attachment proteins. Cell identity markers. These are going to be important when we get to the immune system in AMP2. So think about your immune system, right? It needs to be able to know your cells versus, say, a foreign cell, all right? We don't want foreign cells to get in because they could be harmful. So your immune system kind of learns what your cells look like based on these identity markers. And therefore, if it comes across a cell that doesn't have these identity markers, it knows that that cell must be foreign, and your immune system will, attack foreign cells. So we'll talk more about those in AMP2. All right, that's probably a good place to stop for this video. We're going to start the next video on this slide, where we're going to start to get into specific mechanisms on how things enter or exit the cell. And this is kind of just an introduction, but we'll kind of go through each of these different ways in which things either enter or exit the cell.